Hello boys, welcome back to another session of video tutorials, and this session is meant for class 12th biology students, and this is for 20th February 2021, and this video is especially meant for practicals. At the end of the practicals during your exams, board exam practicals, external examiner will be asking you some questions related to the experiment that you have performed, and also related to that experiment some theory questions also may be asked by the external examiner. So in order to prepare for that, questions posed by the external examiner, we call it YOOC. So we are just preparing you through this module. So the experiment that we are talking about is preparation of temporary amount of onion root tip to study mitosis. And as all of you know, this is slide preparation. It's meant for 5 marks. And the 5 marks are given based on your skill, based on the technique that you use, plus some of the Viva questions. What are those? Question 1. Why mitosis is also known as equational division? The mitosis is also known as equational division because the number of chromosomes in the daughter cell is equal to chromosome number in the parent cell, both 2n, 2n. Why do we use onion root tips or grasshopper larva for studying mitosis? Onion root tips or grasshopper larva are often used for studying mitosis because these tissues have actively dividing cells, and we can easily show up all the stages of mitosis in them. Question 3. Why is early morning considered as an ideal time to harvest the onion root tips for study of mitosis? Again, this is because root tips have active cell divisions in the morning. When we cut such material, we get all stages of cell division in one material itself. Suggest the name of few tissues which are suitable for study of mitosis. We can select the dividing cells of tail of tadpole larvae, epithelial cells of gills of fishes, bone marrow of any vertebrate, root tip cells of herbaceous plant, shoot piece of plants, and of course grasshopper larvae. Why do you prefer monocotyledonous material for cytological studies? Monocot plants are preferable because they have large sized chromosomes which are better visible in under microscope. Which stain is used for studying chromosomes? Already we are using in our lab. Acetocarmine is generally used for studying chromosomes as it stains chromosome deep red while cytoplasm remains unstained. From where do spindle fibers originate? The spindle fibers originate from centrioles in case of animal cells, but in plant cells these originate from the cytoplasm. These are proteinaceous in nature. Question 8. In which stage of cell division chromosomes are seen best? Chromosomes are best seen in metaphase stage. Why? In metaphase, all the chromosomes will be lying in the center of the plate like NCC single file. Okay, that's why. And chromosomes in this stage are very thick and short. That is the thing. Highly condensed form. Question number 9. Suppose a cell has 44 chromosomes. How many chromosomes will it have after mitosis? Of course, equal division, it has to be 44. In mitosis, each of the daughter cells produced have the same number of chromosomes, that is 44, as that of parent cell. Question 10. What do you mean by the term karyokinesis? Karyon in Latin means nucleus. Kinesis means division. Nuclear division is called karyokinesis. Similarly, cytokinesis, cyto means cell, kinesis means division. Cellular division is cytokinesis. During mitosis or meiosis, first it is nuclear division. Karyokinesis will be followed by cytokinesis. Next, what shape would a metacentric and submetacentric chromosome exhibit during the anaphase stage? Of course, type of chromosome, this metacentric shape will be V shape. submetacentric L shape, and acrocentric J shape, and telocentric I shape. Question 12. In which stage do the chromatids start moving towards opposite poles, north pole, south, of course, anaphase. Question 13. Where do spindle fibers attach on the centromere during cell division? Spindle fibers attach themselves to kinetophores during cell division. Next, where does kinetophore consist of, or what does kinetophore consist of? It consists of proteins and specific sections of chromosomal DNA at the centromere. Question 15. How does cytokinesis differ in animal and plant cell? The difference cytokinesis is, in animal cytokinesis it occurs by cleavage. In plants it occurs commonly by cell plate formation. Spindle first is still half cytokinesis, whereas in animals spindle starts disappearing in early telophase stage. A mid-body of dense material is formed at the middle of the cell, whereas mid-body is not at all formed in plant cells. Question 16. Name the chemical which can inhibit cell division during metaphase. Of course, we use that. Colchicine inhibits the formation of spindle fiber during metaphase stage. Name the source of colchicine. It's obtained from plant Colchicium autumnale of the family Liliaceae. Question 18. What is Fragmoblast? It is a cell plate formed in plant cell during cytokinesis. It is formed in the center, which grows outwardly until it divides the cell into two identical daughter cells completely. How chromatin changes into chromosomes? By losing water and supercoiling. The chromatin becomes compact. Why mitosis is called equational division? Because it maintains the same chromosome number both in parental cell as well as in daughter cell. Where do mitotic division takes place? Of course, it takes place in all vegetative parts of the body. Mitosis and plants and animals. In plants, cell wall is present. In animals, cell wall is absent. Centriole is present in plants. Centriole is absent in animals. No astral rays are formed in plants. Astral rays are formed. Why root tips should be cut and fixed in the early morning hours? Root tip cells have mitotic activity during morning hours when temperature is about 28 degrees centigrade. Why root tips are treated with 1N-HCl? For hydrolysis, so that middle lamellae between the cells dissolve. Question 25. Why we select root tips for the above experiment? Of course, just now we said meristematic activity will be in the peak. Why monocots are used? Again we said chromosomes will be large. Name 3 types of meiosis: zygotic, gametic and sporific. And last, what is synapsis? Pairing of homologous chromosomes is called synapsis. What is the significance of crossover? Crossing over. It helps in exchange of characters and significant in evolution of new recombinations. So this is how the examiner may ask you around 5 to 6 questions in the form of Viva OC. So we need to prepare like this. Next.